Good morning. Let's continue chapter 7 and finish this chapter. We finished neuropeptide Y last time, and now we're going to talk about endocannabinoids. Endocannabinoids in our body also can be used as neurotransmitter. That's why we call it endocannabinoids, because they are natural, they are made inside the body. Endocannabinoids can bind to the same receptor that the active ingredient of marijuana, which is THC, can sit on. The structure of endocannabinoid is a short fatty acid. Since it's a fatty acid, it doesn't need to be inside the vesicle. It can be released from the dendrite or cell body without any vesicle. You will see in a few minutes that endocannabinoids, when they are released from the cell body or dendrite, they're going to go back to the presynaptic neuron and block the presynaptic neuron. So they create inhibition of releasing further neurotransmitter from the presynaptic neuron. We will see this in a second. Endocannabinoid has two different receptors called CB1 and CB2. CB1 mainly is in the central nervous system. CB2 mainly works in immune system. There are two important endocannabinoids that you need to know. One is called anandamide, and the other one is called 2-arachidinyl glycerol. Anandamide has another name. It's called N-arachidinyl ethanolamine, therefore is abbreviated by AEA, and 2-arachidinyl glycerol is abbreviated by 2AG. Anandamide, it's very similar to THC that you can find it from plants. It's called phytocannabinoid. It's phytocannabinoid because it's found in the plant. And 2AG, it's very similar to another phytocannabinoid that's called cannabidiol. Endocannabinoid can affect your appetite. Specifically, is 2AG that affect your appetite and also is going to affect your immune system and can manage the pain. Let's say in this example, this is a presynaptic neuron and this is a postsynaptic neuron. Let's say this neuron is going to release a neurotransmitter, and when this neurotransmitter is going to sit on the postsynaptic neuron, it's going to stimulate this neuron, and this is a neuron that releases endocannabinoid. When endocannabinoid is released, it's going to affect the presynaptic neuron. Presynaptic neuron, as you can see, has a receptor for endocannabinoid, which is CB1. This product, AEA or 2AG, when they sit on the CB1, they're going to block the calcium entrance to the cell. So calcium influx to the cell is going to get blocked. When the amount of calcium decreases inside the cell, obviously the cell is not going to release any neurotransmitter anymore. The effect of endocannabinoid is going to regulate the release of neurotransmitter. If this neurotransmitter is inhibiting neurotransmitter, then by releasing endocannabinoid, we're going to block releasing of this inhibitor neurotransmitter. In other words, the cell itself can be stimulated. This idea is called depolarization-induced suppression of inhibition, or it's called DSI. Let's review one more time. If this neurotransmitter is inhibitor neurotransmitter, if by any means you stimulate the postsynaptic neuron and make this cell to produce cannabinoid, this cannabinoid is going to block this cell, and as a result of blocking this cell, it's going to inhibit the release of this neurotransmitter. And because of that, this cell will be even more stimulated. This idea is called depolarization-induced suppression of inhibition, or DSI. Cannabinoid, we said, also is going to increase appetite, and this is a well-known effect of cannabinoid. However, the dilemma is on learning and memory. Let me explore this one for you. In the lab, if you knock out CB1 receptor of a mouse, you will see that mouse is going to show improvement in memory. That shows that stimulation of the CB1 receptor is responsible for removing old memories or aversive memories. However, it can affect any type of memory. On the other hand, in another experiment, it's been reported that when they treated a mouse with a high dose of cannabinoids over a length of few weeks, they noticed that hippocampus, which is the center for new memory, increased the number of the neurons. In another word, neural growth happens in the hippocampus after treatment of the mouse with high dose of THC. So as you can see, the result of cannabinoid on memory is not clear and needs more investigation and more work. So For now, I want you to understand that there are contradicting results on the effect of cannabinoids on learning and memory. The next neurotransmitter that we need to talk about is nitric oxide. Nitric oxide is a gas. This neurotransmitter is made from amino acid arginine. The enzyme that you need to know is called nitric oxide synthase. Since it's a gas, we don't need any vesicle. It can release from the cell membrane and leaves the cell membrane, comes to presynaptic cleft. We create NO as needed, so there is no storage for that either. NO use a second messenger, and that second messenger is not cyclic AMP but is cyclic GMP. Remember, we always talk about cyclic AMP as a second messenger. You have to understand there is another second messenger. It's called cyclic GMP. Do you remember the enzyme that makes cyclic AMP is called adenyl cyclase? The enzyme that makes cyclic GMP is called guanyl cyclase. NO not only is a neurotransmitter, but NO can be a paracrine. Do you remember the term paracrine? If this is a cell, release a chemical, that this chemical is going to affect other cells. This action is called paracrine. Paracrine is a process in which one cell is going to create a chemical that affects neighboring cells. Please compare this one to hormone that is released from some cells, is going to get to the blood and is going to affect distant cells. So hormone is going to affect distant cells. Paracrine is going to affect neighboring cells. NO can be a paracrine in the GI system and in the respiratory system and in the blood vessels. Also, I want you to know that NO is going to help killing bacteria in the wall of the smooth muscle of the blood vessel. NO is going to get released and it's going to stimulate guanyl cyclase to create cyclic GMP. You will see this cyclic GMP at the end is going to cause a smooth muscle of the vessel to get relaxed. As a result, it's going to create vessel dilation. In fact, that's the way that Viagra works. Viagra is going to make you to produce more NO. NO is going to get released, creates more of cyclic GMP, and at the end, this one is going to create dilation of the blood vessels in the penis and cause erection. NO as a neurotransmitter can become a retrograde inhibitory neurotransmitter. In other words, inhibits presynaptic neuron. When NO is released, it's going to increase the amount of cyclic GMP inside the cell, and as a result of that, can make smooth muscle relaxation. In the case of vessels, relaxation of the smooth muscle of the blood vessels cause vessel dilation. That's the reason that Viagra causes erection. Viagra is going to increase NO. NO is going to increase cyclic GMP. Smooth muscle of the blood vessel is going to get relaxed. Blood vessel is going to get dilated. More blood is going to come to the vessels in the penis and cause erection. With the same token, NO can be released in the smooth muscle of the respiratory tract and relax the airways, and NO can help killing bacteria. As a neurotransmitter, remember NO can do retrograde inhibition. In other words, can go to the presynaptic neuron and inhibits presynaptic neuron. Another neurotransmitter that is a gas is carbon monoxide. I'm sure you know that carbon monoxide is a toxic gas in a large concentration. If you have too much carbon monoxide in your body, it's going to attach to the heme and prevent heme to carry oxygen. Let's take a look. This is your RBC. Inside the RBC, we have some hemoglobin. This is supposed to be hemoglobin. Hemoglobin can carry oxygen. If you have too much CO, CO is going to attach to hemoglobin. As a result, hemoglobin will not carry oxygen. So therefore, CO poisoning is going to make you to have cyanosis. Cyanosis is due to lack of oxygen. You may ask me, where does CO is going to come from? Well, in a normal person, the CO is going to come from breaking down hemoglobin. When you break down hemoglobin, you're going to create a pigment. It's called biliverdin. And as part of this process, you're going to create some CO. That is okay. However, when you get poisoned by CO, the CO is going to attach to hemoglobin and prevent hemoglobin to carry oxygen. In the normal amounts, the CO would be beneficial. This CO can act as a neurotransmitter. Remember a few minutes ago I told you that hippocampus is part of the brain that creates new memories? CO as a neurotransmitter can cement this memory in the hippocampus. It was noticed that if we block carbon monoxide, we erase the memory that we already created. The memory is going to get lost. There's also suggestion that CO may protect your brain against excess of neuronal activity. In other words, it's going to decrease the firing of neurons. Well, this needs more investigation, but for sure CO deals with memory. I'm sure you remember the enzyme guanyl cyclase, or it's called guanylyl cyclase. This is the enzyme that creates cyclic GMP. This enzyme also contains heme. By attaching CO to heme in the guanylyl cyclase, this enzyme becomes active and is going to create cyclic GMP. It is noticed that those neurons that contain this enzyme, it's only in a specific area of the brain. Which neuron contain guanylyl cyclase? The answer is olfactory nerves, they have guanylyl cyclase inside. Some cells inside the hippocampus, which are called pyramidal cells, also they have guanylyl cyclase. And finally, we have some neurons in cerebellum, which we call them granule and Purkinje cells. Those cells, those neurons, also they have guanylyl cyclase. So if I ask you, which neurons are target for CO, the answer is those neurons contain guanylyl cyclase. Olfactory nerves, pyramidal cells in the hippocampus, and granule and Purkinje cells of the cerebellum. Remember, hippocampus is for learning and memory. As a result, we can say CO can improve our memory. As a last note, there is inhibitor of carbon monoxide, and that's called zinc protoporphyrin 9. Zinc protoporphyrin 9 can block the effect of carbon monoxide. Let's move on to the next neurotransmitter, and that is ATP. As you know, ATP is adenosine triphosphate. As a reminder, let's talk about ATP and its structure. I'm sure you all remember that we have different bases. Those bases that they have two rings is called purine, and the one that has only one ring is called pyrimidine. We have adenine and guanine as a purine, and we have uracil, thymine, and cytosine as pyrimidine. ATP is adenosine triphosphate, a combination of the adenine with 3-phosphate and a sugar is called ribose. ATP can be a neurotransmitter, but also adenosine can be a neurotransmitter. Adenosine is an adenine and sugar without phosphate. Usually ATP or adenosine, they are being a co-transmitter. In other words, they don't release alone. They release with other neurotransmitter like epinephrine, like acetylcholine, like serotonin. So they go with another neurotransmitter. Since ATP and adenosine, they are coming from adenine, and adenine is a purine, we have two receptors for purines. We call P1 and we call P2. P1 is a receptor for ATP and ADP, and P2 is a receptor for AMP and adenosine. As we said, ATP or adenosine, they are co-transmitter. For instance, if you release ATP with noradrenaline or norepinephrine, that is going to stimulate constriction of the blood vessels even more. Remember, norepinephrine is going to constrict blood vessels, but if you release ATP with norepinephrine, this constriction is going to get even stronger. Or you can release ATP with acetylcholine. Acetylcholine is going to cause intestinal contraction. If you release ATP with acetylcholine, intestinal contraction is going to be even stronger. ATP or adenosine can be also released from non-neural cells. They can become paracrine regulator in the blood clotting process, in taste, and also as part of the pain mediator. As we said, ATP and adenosine are co-transmitter. We said they can be released with norepinephrine, or they can be released with acetylcholine. They can also be released with substance P, or with VIP and NO. In the autonomic nervous system, if you remember, autonomic nervous system is a nervous system that goes to your heart, goes to your glands, and goes to your smooth muscle. In the autonomic nervous system, we have some neurons that they don't release norepinephrine or acetylcholine. Those neurons that they don't release norepinephrine or acetylcholine is called NANC, non-adrenergic, non-cholinergic. Majority of this neuron is found in the GI system. You will learn later those neurons in the GI system that they don't release norepinephrine or acetylcholine, they release VIP or NO instead, and with those two neurotransmitters will also release ATP. So ATP can be a co-transmitter with VIP and NO in non-adrenergic, non-cholinergic neurons. That is going to bring us to the end of neurotransmitters. Before we move on from this one, I want to remind you that there are so many neurotransmitters, and the combination of these neurotransmitters is going to create who we are, and as a result, you can see this can be quite complex because each of these neurotransmitters can affect other neurotransmitters. Manipulation of one can affect the whole system and create a lot of side effects. That's why it's not that simple to increase or decrease one neurotransmitter and expect that only one specific effect happens in our body. So now let's move on from neurotransmitter and talk about synaptic integration. I want to take a look at this picture. Let's say here is a neuron, and there are three different axons comes and affect this neuron. I want to remind you that although we talk about one neuron attached to another neuron, but you know, in reality, in our body, this is not going to happen. In other words, you don't have one neuron attached to another neuron. In majority of time, you have multiple neurons that affect one neuron. So in this picture, you can see three neurons are affecting this neuron. What's the final response of these three? Well, the answer is they're going to summate. This type of summation is called spatial summation. For instance, if this neuron by itself cannot bring this cell to the threshold, then we need to use other neurons. Combination of these three neurons, let's say they can bring this cell into threshold and create action potential. We're going to summate the effect of these three, and finally this cell is going to fire. What if one of these neurons are inhibitory and two of them are excitatory? It's the same thing. You have to look at the overall effect of these three. If the effect of these two against this one, it still brings the cell to the threshold, it's going to fire. But if this guy is strong enough to block the effect of these two, as a result, by the time we get to axon hillock, we don't get to the threshold, then this neuron is going to get blocked. So summation is adding negative and positive and see what the final effect would be here. This type of summation is called spatial summation. We have another summation that is called temporal summation. What is temporal summation? Well, think about that this neuron is going to affect this neuron and try to make this neuron to fire. But one stimulation of this neuron to this neuron is not going to bring this cell to the threshold. As a result, if this cell send impulse back to back, if the frequency of sending impulse of this neuron increase, as a result produce lots of neurotransmitter here, and as a result this cell becomes stimulated back to back, then there's a chance for this cell to go to the threshold. So one impulse may not bring this one to the threshold, but multiple impulses of this one may bring this one to the threshold. This type of summation is called temporal summation. This picture shows spatial summation again. As you can see, the first neuron is going to create excitation, but is not big enough to bring to threshold. But when you add the second one, then 1 and 2, they're going to combine together and get to the threshold. So this is a spatial summation. As you can see, each one by itself cannot get to threshold, but two of them combined is going to get to threshold. This is also spatial summation. As you can see, one cannot get, but two of them together is going to get to threshold. So let's change the gear and talk about synaptic plasticity. Synaptic plasticity is the ability of a synapse to become strengthened or weakened over time. What does that mean? Sometimes when you use one circuit, that circuit becomes stronger. Let's say if you put a mouse inside a maze, and at the end of the maze there's food, the mouse is going to try to find a way to get to the food. At the beginning, it's going to take him a while. If you do it second time, it's going to go faster. And the third time, it's going to go faster. Every time you put the mouse at the beginning of this maze, it's going to get to food faster and faster. Why is that? Because the brain of this mouse creates synaptic plasticity by increasing the activity. It's going to get improved every time. In other words, neurons, they're going to create a map. They know exactly what should be done next. So that's a positive synaptic plasticity. Sometimes by reusing a circuit multiple times, instead of getting improved, it's going to get decreased. It's going to get weakened. So remember, synaptic plasticity is not always going to increase the activity. Sometimes it's going to decrease the activity. If this increase or decrease of the activity happens in a very short time scale, we use the term synaptic facilitation for improvement and synaptic depression for weakening. I don't want you to think that using one circuit multiple times always going to create facilitation. Sometimes it's going to create depression. This term is used when we use synaptic plasticity in a very short period of time. Why synaptic facilitation? Because as a result of using that circuit over and over, you're going to increase intracellular calcium, and as a result, you're going to release more neurotransmitter. But why synaptic depression? It seems like the most common reason for synaptic depression is lack of neurotransmitter. When there's not enough neurotransmitter, then the cell is going to start decreasing the activity. Let's take a look at this example. Here is one neuron. It's going to affect this neuron. As you can see, sensory neuron is going to stimulate and the cell is going to respond. And if you stimulate again, there's going to be a synaptic depression. Why is that? Well, because when the first time you stimulate this neuron is going to release neurotransmitter. However, by second stimulation, there's not enough neurotransmitter to be released. As a result, it's going to create synaptic depression. Take a look at this one. When you stimulate this neuron for the first time, then you're going to release calcium. If you stimulate this neuron right after, before this calcium is being reuptaked, so the calcium inside the cell is going to go even higher, it's going to make you to release more neurotransmitter. So that is going to create a synaptic facilitation. If this happens in a longer time, we use the term long-term potentiation or long-term depression. Long-term potentiation is the way that you create long-term memories. When you stimulate a neuron multiple times over and over, then soon this neuron is going to remember the signal and it's going to lay down a memory of that signal. I don't think we need to discuss long-term depression and why this one happens. Now let's change the gear and talk about synaptic inhibition. This is something you already know. We talked about this one. By releasing glycine or GABA, you may inhibit postsynaptic neuron. GABA and glycine can hyperpolarize the second neuron, and as a result, the chance for the second neuron to get to threshold will decrease. If, let's say, neuron 1 inhibits this postsynaptic neuron, even though second neuron try to stimulate, but because of the inhibition by the first neuron, you are not going to get to the threshold. That's a synaptic inhibition. Now, we already know this one. I explained this one again because I want to talk about presynaptic inhibition. So what is presynaptic inhibition? Let's take a look at this picture. This is a neuron, axon number 1. You can see this axon number 1 is going to make a synapse with axon number 2. Imagine this axon number 2 trying to stimulate this neuron. But axon number 1, before even this axon number 2 get to this cell and stimulate it, is going to be inhibited. That is called presynaptic inhibition. Now realize there's a difference between this one and this one. Neuron number 1 and neuron number 2, they have no connection here. 1 and 2 separately is going to attach to postsynaptic neuron. Here, axon number 1 blocks axon number 2. So this is called presynaptic inhibition because it's going to inhibit axon number 2 before even axon number 2 stimulates postsynaptic neuron. In this scenario, axon number 1 is going to prevent influx of calcium. As a result, axon number 2 does not get a chance to release excitatory neurotransmitter. An example of this one would be when axon number 1 release opioid and prevent axon number 2 to release substance P. Let's say axon number 2 is the one that is supposed to release substance P to stimulate this one to go to the brain and you feel pain. If axon number 1 release opioid is going to come and sit on the axon number 2 and prevent axon number 2 to release substance P. That's one way that opioid is going to prevent feeling pain. And finally, let's talk about neuromodulator. So far, we talked about one neuron is going to release neurotransmitter to affect another neuron. Either in this case or in this case, one neuron is going to release neurotransmitter to affect this neuron or this neuron. What if one neuron is going to release a neurotransmitter or chemical that affects many, many neurons? It's going to create a very massive result. In this case, we call it neuromodulator. When a neuron releases one or more chemicals to regulate diverse population of neurons, that is called neuromodulator. This is so different than synaptic transmission that we talked. One neuron is going to release neurotransmitter to affect another neuron. Example of neuromodulator would be dopamine, would be serotonin, would be acetylcholine, would be histamine or norepinephrine. The same neurotransmitter that we talked about as a neurotransmitter that releases from one neuron also can act as a neuromodulator. And that is going to bring us to the end of this chapter. We're going to talk about chapter 8 very soon.